Well, welcome everybody to today's Leukemia Care education webinar with the purpose to update on latest treatments and developments in myeloproliferative neoplasms, tongue twister for me, MPN, it's probably easier for me to say. A little bit of housekeeping just before we start. Those who've attended webinars before will note that you're not able to share camera or speak directly, but we do encourage you, if you can, to use the chat function at the bottom of your screens to post any questions, introduce yourself, say hi to everybody. This event will be recorded, is actually being recorded, and the recordings will be made available on the website probably now after the weekend, so that you can revisit, because no doubt Professor Mead's got plenty of information and slides to share, and you can revisit those at your own leisure. We're also streaming live to an audience on Facebook, so welcome to everybody on Facebook. Similarly, I have a colleague with me who will collect any questions that you might have. So if you have any questions, just pop them below in the discussion thread underneath the video, and my colleague Lydia will pick those up and get them to us for Adam to answer in the Q&A session after his talk. Yeah, we're assuming that the majority will have some basic knowledge of the condition and issues, but if you're unsure of anything or don't understand anything that Adam talks about, please pop a note in chat, and Adam will see that on his screen and address that during his talk. So if I introduce myself first, my name's Nick. I work for Leukemia Care in the advocacy team. And I think at the moment I've got nothing further to really give you other than to introduce you to our speaker for the day. Adam, please, could you introduce yourself and share your talk?
Yes. Hello, everyone. My name is Adam Mead. I'm a haematologist in Oxford, where I also run a research group. And my particular area of interest is myeloproliferative neoplasms. And so my remit for today is to give you an update on the kind of hot topics that came from the American Society of Haematology meeting in December 2022. So hopefully, with a bit of luck, you can now see my
Adam,
script.
you're coming across quite a little faint. I don't know if there's any way you can raise your volume at your end.
How about if I speak at this volume? Is that better?
I know you've got a sore throat, so I don't want to stress your throat, honestly.
Yeah.
But yeah, that's fine. We can hear you.
Yeah. So apologies, everyone. I've picked up some form of lurgy from my children, most likely. But I'll see how things go. So my remit was to talk about hot topics coming out of the American Society of Haematology 2022 meeting in December, and there were a lot to get through. But the overarching kind of focus, I think, of some of these areas of clinical research I'm going to talk about is how many of the efforts are moving towards what we call disease modification in myeloproliferative neoplasms. So that's what I want to talk a little bit about today, just to explain what we kind of mean by that term, and then how the research is going. So by way of background, myeloproliferative neoplasms are chronic forms of blood cancer. They are quite rare diseases, affecting, about 1 in 100,000 people per year for each subtype. But the fact is, people live with these conditions for many years. So the prevalence, as in the number of people living with these conditions in the UK, is quite high. It's estimated about 60,000 people living in the UK with a myeloproliferative neoplasm. We know the chronic forms of the condition, essential thrombocythemia, high platelets, and polycythemia vera, too many red blood cells, are particularly associated with an increased risk of blood clots, and also an impact on quality of life due to disease-associated symptoms that can affect people in many different ways. As the disease progresses and gets more advanced, bone marrow scarring can occur in a certain proportion of people. That's a more advanced form of blood cancer associated with shortened life expectancy. And in some people, the disease can progress to a very nasty form of leukemia. Thankfully, that is a rare event, but it can happen, and we need to learn about how to better treat this condition, but also how to prevent it from happening in the first place. So we do have some therapies available for these conditions already that are quite effective at controlling some of these risks, controlling the blood counts, improving the enlarged spleen that particularly occurs in patients with myelofibrosis. But there's definitely a need for new therapies and new ways to measure how the therapies are working. So what I put on this kind of cartoon is an explanation. You may hear this term more and more, which is disease modification. And I just want to talk a little bit about what I think that means. You might hear different opinions about what that means, but I'll tell you what I think it means. And that's summarized in this cartoon here. So if you think of blood cell production as essentially occurring in the bone marrow, you have a group of normal, all of us have about 100,000 normal hematopoietic stem cells that throughout our lifetime produce blood cells, millions of blood cells every minute of every day throughout our lives. And what happens during the development of MPN is one of these cells, probably many decades before the disease develops, picks up a mutation. And the classic mutations you may well have heard of is the one in JAK2. So a JAK2 mutation, but it takes a long time for that one cell to grow, as shown by the cells in green, to the extent that it actually causes a problem for someone. And that's why you may hear this term clonal disorder. So clonal means that it's all derived from a single cell. So it's cloned from a single cell. And over time, in some patients, you can get additional mutations in addition to the JAK2 mutation, and that can cause the more advanced forms of myeloproliferative neoplasm. And what I regard disease modification as is essentially reversing this process. So returning the abnormal bone marrow back to normal hematopoietic stem cells that produce normal blood cells. And it seems obvious to me that if we can achieve that, that will correlate with improved symptoms and improved outcomes for patients. And one of the things that I'm going to talk about quite a lot today is how in many of the clinical studies that were presented at ASH, the investigators are using this term molecular response, which is a surrogate marker of what I'm putting in this cartoon. Basically, it's measuring the number of cells that have the JAK2 mutation. So the number of cells that are green versus blue. And if you can reduce the level of the JAK2 mutation, what that means, what's happening inside the bone marrow is a return from this type of situation back to this type of situation when you've got normal blood cells. So, that kind of what we call disease modification is clearly a goal. Obviously we want to return the bone marrow back to normal function. But is it achievable? That's the question. And I think there's a few lines of evidence coming from the recent studies presented at ASH that are reasons to be optimistic. So the first study I wanted to present is one that I was helped to lead, but Claire Harrison was the chief investigator of this study called the MAGIC study. And this was a study that ran in the UK. In fact, we first started planning this study back in 2010, which tells you how much time and effort goes into developing and running a study of this nature. So what this study tested was patients with polycythemia vera, one of the chronic forms of myeloproliferative neoplasm, and patients who were not responding as well as we would like to hydroxycarbamide, the standard treatment. So they were either intolerant, getting side effects, or resistant to that treatment. And in those patients, they were randomized to the best available therapy that we currently have versus treatment with the JAK2 inhibitor, ruxolitinib. This group of patients, particularly with this hydroxycarbamide resistance or intolerance, is associated with quite adverse outcomes. So it's a group of patients with a definite unmet need for new treatments. And I'm going to cut to the chase with the results of the study. The study design is summarized here. So patients were randomized to best available therapy or ruxolitinib. So this is a standard randomized trial design, 1-to-1. So it was an equal ratio. And one of the things that we did in the study was we very carefully collected samples throughout the study. And that proves, if any of you are taking part in clinical studies, you will know that often there's quite a lot of sample collection. And that is so important because it really increases the amount we can learn by running these studies. So the outcome of the study was really very clear-cut, that JAK2 inhibitor treatment with ruxolitinib was superior to best available therapy in this situation. More patients achieved a complete remission, so normalization of their blood counts. But perhaps more importantly, what we call the event-free survival, that's the chance of being alive and not suffering from any complications of the disease, such as a blood clot, progression to more advanced disease, or a bleeding event. And here you can see almost the rate of those events is halved for patients on ruxolitinib compared to patients on best available therapy. So a very clear-cut outcome. What the trial also showed us is that, often when patients with polycythemia or other forms of MPN come up to clinic, we focus on the blood counts. Have we managed to control the blood parameters as well as we would like? And what this study showed is that it's very, very important to achieve good control of those blood parameters. So those achieving perfect control of the blood parameters, so a complete remission, had markedly improved outcomes compared to those that did not. So a very positive result for ruxolitinib in this disease setting. And the other important thing that I wanted to point out is that, through the sample collection that we carried out on this study, we could carry out a detailed molecular analysis. That's what I was explaining at the beginning, measuring the amount of the JAK2 mutation in the blood cells. And what that shows us is that, let me just see if I can, I don't know, can you see this little box here? Is that in the way of the slides, or is that just on my slide?
It's just off to the left of the slide. It's now in front of the slide.
Yeah, I need to try and get rid of that because I can't see my own slides. Let's see. There we are. That's better. Is that better?
That's better. It's not on the slide itself.
Yeah, good. So what that showed us is that ruxolitinib was far more effective at making the level of the JAK2 mutation go down. In some patients, the JAK2 mutation disappearing entirely. What we could also do, using some cutting-edge techniques that we've developed in Oxford, in my laboratory, we could look at the level of JAK2 in the stem cells from the bone marrow of these patients. So not only were we clearing the JAK2 mutation from the blood cells, but also from the stem cells in the bone marrow. Those are the crucial cells that propagate the disease. So this is, I think, really good hard evidence of disease modification. We're improving important endpoints for patients in terms of reduced rate of complication of the disease. And in parallel, we're making the JAK2 mutation go away and clearing the disease-associated stem cells so that they, in some patients, were no longer detectable. And that was work done by all the people shown here, from team from London, Cambridge, and Oxford. Is it important to achieve those kind of molecular responses? Well, that is shown here. Patients that achieved a molecular response had far fewer complications of the disease, as shown here in purple, than patients that did not achieve a molecular response, shown here in the kind of orange color. In terms of complications of the treatment itself, there were some important things to note with the ruxolitinib treatment. Ruxolitinib is a JAK2 inhibitor, so it's JAK2 and JAK1 that it inhibits are important for the function of immune cells. So it is immune suppressive, and it's a well-recognized phenomenon that patients are at increased risk, particularly of shingles, so chickenpox virus reactivation when they're on the treatment. So that's something that needs to be carefully looked out for. So in summary, the MAGIC PV study, a really long-term endeavor in the UK with 190 patients in the UK taking part, confirms and challenges the current treatment approach for patients. It confirms by way of the fact that we should be aiming to get the blood counts under good control, but also challenges the current therapeutic algorithm in that it really highlights that we should be trying to use ruxolitinib in patients in this situation, so polycythemia vera who are resistant to hydroxycarbamide treatment. The challenge is that ruxolitinib treatment in this situation is not currently funded in the UK. If you were in Scotland, it is funded. If you're in England, it's not. So that's a rather unsatisfactory situation that we hope to resolve. So another reason why it's so important to run clinical trials and for people to take part in clinical trials, because the MAGIC study has created a data set that is forming the basis of a submission to the National Institute of Clinical Excellence, which we hope will result in funding of ruxolitinib being made available for patients on the NHS with a bit of luck, if it all goes well, later this year. So studies from the UK having really huge impact, both for UK patients and internationally. The follow-up from that study is this study, which is currently open in the UK, which is called the Mithridate study. This is now asking the question, well, if ruxolitinib is such a good treatment in patients who are resistant to the standard treatment, is it a better treatment than the standard treatment itself? So as in a first-line treatment. So that's what this trial is asking, patients with newly diagnosed or untreated polycythemia vera, or patients who are on treatment and do not have signs of resistance. So the summary is shown on this slide here, the kind of high-risk patients we want to include. And then again, it's a randomization between ruxolitinib versus best available therapy treatment, which can either be hydroxycarbamide or interferon. So really challenging the treatment paradigm and asking whether ruxolitinib might be useful as a first-line treatment. And this is to pass on thanks to particularly the patients that took part in the study and all of the investigators. So there were a number of other people that looked at a similar question relating to JAK2 molecular response in patients treated with ruxolitinib. And the bottom line is, everyone is now starting to see the same thing, that patients that achieve a substantial molecular response, or a reduction in the level of the JAK2 mutation, although it's not the majority of patients at the moment, those patients that do achieve such good response have got dramatically reduced risk of developing complications of the disease versus those patients that do not achieve those kind of molecular responses. So should this lead to an immediate change in practice is a reasonable question. And I think at the moment, no. And I say no because what we don't know is which response is more important. I've shown you the data that suggests controlling the blood counts is really important, and a molecular response is also really important. We don't know which is more important. And, that requires some more careful thought, and we need to make some national guidance on that. But I think as a minimum, as clinicians and as patients, we should be thinking about measuring the JAK2 level response to treatment, because it's a very useful marker of how patients are responding to the treatment and their risk of developing complications of the disease. Okay, so if we move aside from the JAK2 mutation and think more about calreticulin mutations, so this is a mutation that occurs in about 30% of patients with ET, and it is a mutation that is mutually exclusive with JAK2. So in patients with ET that have a JAK2 mutation, they don't have a CALR mutation, and vice versa. And in essential thrombocythemia patients, both JAK2 and calreticulin mutant, the Imago have been running this study of bomedemstat, which is a tablet treatment, an oral treatment that is very effective at controlling the platelet count. So this is, essential thrombocythemia is a condition characterized by a very high platelet count, and you can see the dramatic data on the study showing how effective this treatment is at normalizing the platelet count. So a very exciting new treatment coming through for patients with essential thrombocythemia, and also effective at controlling symptoms for patients. And that particular agent is being taken forward in a phase 3 study, so the randomized type of study. And this particular agent has also been bought by another company now, who are going to put a lot of investment, I think, in the development. So lots of potential new therapies coming through. So how about other treatments that are coming through for essential thrombocythemia? Well, one possible treatment is this drug called pelabresib. More about that later in myelofibrosis, but it is also a drug that has been used successfully as a single agent in persons with essential thrombocythemia. So new treatments coming through for polycythemia vera, new treatments coming through for essential thrombocythemia. What about myelofibrosis? So in myelofibrosis for almost 15 years now, not quite, but about 12 or 13 years, we've had JAK2 inhibitor treatment available for patients with myelofibrosis in the UK, which has had a dramatic impact on outcomes for patients. But nevertheless, not all patients respond as well as we would like, and we should always be thinking, how can we do better? In particular, how might we start inducing these types of disease modification that I talked about in the beginning. And I want to highlight some studies that are using new treatments in combination with the ruxolitinib treatment. So ruxolitinib is the standard treatment, excuse me, and I'll show you some data about a combination treatment that's being tested, adding in the other standard treatment that is often used in MPN, interferon, but asking how about using them both in combination. And then I'll show you some data on navitoclax in combination with ruxolitinib and pelabresib, a BET inhibitor, in combination with ruxolitinib. Excuse me. So parsaclisib was a drug that was in clinical development. Excuse me.
Yeah,
and
okay. Need to take a break for a second.
Well, I'll just have a little drink. Sorry, and typical that I got the lurgy before doing this.
Yeah, I'm aware that you're pushing yourself there as well.
Okay, and parsaclisib, which is a PI3 kinase inhibitor. Unfortunately, the drug company Incyte has stopped the clinical development of that particular agent, but there's still a lot of exciting drugs in development in this disease area. So I'll skip that slide in the interest of time, and we can talk about, first of all, the combination treatment with ruxolitinib and interferon. Two standard treatments available for MPN patients in the UK, and a French team led by Jean-Jacques Giladian asking the question, well, what about using them in combination? And the results are quite striking. The spleen response rates, this is often used as the kind of measure of response in myelofibrosis patients, occurring in as many as 70% of patients. That is an exceptionally high level of response. And what about if we look at these molecular responses, thinking about, are we inducing disease modification? Well, the answer seems to be for patients who've just got a JAK2 mutation, it looks like that this combination treatment is inducing molecular responses. Unfortunately, less so for patients with calreticulin mutation or MPL mutation or patients with additional mutations. The evidence of molecular responses was less convincing. But I'll come back to some very exciting new approaches in development for calreticulin mutation patients later. So this particular combination with interferon and ruxolitinib looks potentially quite interesting, particularly for patients with myelofibrosis who just have a JAK2 mutation. What about this other agent called pelabresib, what's called a bromodomain inhibitor, pelabresib? So again, that has been used in combination with ruxolitinib in patients who have not previously been treated. So bringing this type of combination treatment to the first line. And at ASH, thinking about disease modification, these data were presented showing that, you know, there are some patients achieving a molecular response with this combination treatment that I think is potentially quite exciting. And this particular combination treatment is being taken forward again to a phase 3 study. The other, I think, very exciting combination treatment, if you think about the three, so interferon with JAK2 inhibitor, I've told you about. Pelabresib with JAK2 inhibitor also looks interesting. This treatment, navitoclax, made by the drug company AbbVie in combination with ruxolitinib, also looks particularly interesting. In particular, you know, patients are achieving very high rates of spleen volume responses, but very excitingly, I think, a significant proportion of patients are achieving these kind of molecular responses, even after just 12 weeks on treatment. Potentially could be a game changer, this treatment. And it's very exciting, I think, to know that we'll be getting the results of the phase 3 study coming through later this year. So I'm very excited about that one. And, you know, what they also showed is that those patients achieving a molecular response had much better outcomes than patients that did not. Again, justifying that maybe we should be rethinking some of the treatment goals in myeloproliferative neoplasm towards trying to achieve these types of molecular responses. So one interesting thing that often comes up as a point of interest in terms of measuring the response is whether reversing bone marrow fibrosis in myelofibrosis is an important thing. You know, the disease myelofibrosis is diagnosed on the basis of the presence of bone marrow fibrosis. So it would be reasonable to ask the question whether it's very important to reverse that. And that's something that Ruben Meza asked in this study using some data from the Simplify-1 study, which was a study of momelotinib, a JAK2 inhibitor, versus ruxolitinib, the other standard JAK2 inhibitor, as a first-line treatment. And the bottom line is, what that study showed is there was no difference in survival, spleen response, or symptom response in patients that achieved a reversal in their bone marrow fibrosis compared to those that did not. So the take-home message from that is maybe reversing the bone marrow fibrosis is not as important as we might have thought, and we should maybe focus on other endpoints that are important for patients: symptoms, improving anemia, and molecular responses, because we know that reduces the risk of disease complications. Okay, so I've talked about some of those issues, so I won't go over that slide again. And I've talked through these studies, so I won't go over that again. Okay, just some kind of points for update, particularly for persons with myelofibrosis in the UK. The first thing is that we're likely to get access to this new JAK2 inhibitor, momelotinib, which it's a similar drug to the available drug ruxolitinib, but works in a slightly different way through this target, ACVR1. That alters the way the body metabolizes iron, and by doing that, it can improve levels of anemia. So one of the problems we encounter with ruxolitinib treatment is that it often causes anemia in patients with myelofibrosis rather than improves it. So I think momelotinib may be a very exciting new treatment that is available from the drug company on a compassionate use basis from now in the UK. Okay, I will skip over that. And the other drug that is coming that is interesting is pacritinib, another JAK2 inhibitor treatment. Pacritinib is interesting because it does not inhibit JAK1, and it's very useful in patients that have a low level of platelets. Many of the other JAK2 inhibitors we cannot use in patients with low platelets, whereas pacritinib seems to be a drug that is particularly amenable to the use in patients with low platelets because it does not make the platelets drop any further. And even in patients with low platelets, we can see good levels of spleen volume response in patients receiving pacritinib versus best available therapy. And there is a trial running in the UK currently called the Pacifica study, which is specifically for patients with myelofibrosis with a platelet count less than 50. Okay, as if that weren't all enough information and probably too much to take everything in, but as you've heard, the slides will be available if you want to recap anything. This for me was the most exciting presentation at the ASH meeting. I'm going to go through it pretty quickly, but just so we got time for questions at the end. But I wanted to highlight it because this was presented at the plenary meeting at ASH. So the plenary meeting is the plenary session is out of 5,000 abstracts presented, submitted to the American Society of Haematology. The top 6 abstracts, the most exciting, are presented at the plenary session. And this was one of them, the development of a new antibody treatment for patients with myeloproliferative neoplasm and mutant calreticulin. So the thing that you probably don't know about calreticulin mutations is that it's a very unusual example in a blood cancer, or any cancer for that matter, of a mutation that causes the disease that is actually expressed on the surface of the cells. And that is important because it makes it amenable to targeting of that abnormal mutant protein with an antibody. So it's a rare example of a disease-specific protein expressed on the cell surface, providing a unique opportunity to selectively target the blood cancer clone, leaving the normal cells intact, because the normal cells do not have mutant calreticulin. So if you can make an antibody that just attacks the cells with the mutation and not the normal cells, that should in principle be a very powerful way to eradicate the blood cancer cells selectively whilst leaving the normal cells intact, and therefore hopefully inducing disease modification, shown in the cartoon. There's the antibody. This is an antibody shape, a classic antibody shape, attacking the mutant calreticulin. So that's what these folks at the drug company Incyte did. Incyte is a company that's particularly interested in myeloproliferative neoplasms because they developed the JAK2 inhibitor ruxolitinib, which has been a huge success. So they developed this antibody that selectively targets the mutant calreticulin protein. And lots of data here, lots of jargon. Don't let that faze you. I will just explain it in really simple terms. The blue line is the antibody treatment. The green line is a non-specific antibody. And this is the amount that the antibodies are inhibiting cell proliferation in a model of myeloproliferative neoplasm. And you can see the therapeutic antibody hugely effective at inhibiting cellular proliferation. and it's very selective for mutant calreticulin. For mutant JAK2, it doesn't work, so it only works for the mutant calreticulin. But as I said earlier, we've got other treatments that are looking very exciting for mutant JAK2, including interferon. Here's another way of looking at this using patient samples. So these are patient stem cells, again showing that the therapeutic antibody is very strongly inhibiting the abnormal cell signaling compared to normal cells or cells carrying the JAK2 mutation. And here's an assay of stem cells, again showing that the therapeutic antibody having a dramatic effect on the number of MPN stem cells, but only selectively in those carrying the mutant calreticulin, not the JAK2 mutation. They also showed that it was highly effective at controlling platelet count. Here's a mouse model of abnormal platelet counts, very high platelet count, return to normal with this antibody treatment. Really dramatic effects. And here's what the bone marrow looks like in this mouse model. You can see in brown, these are the megakaryocytes. These are the cells that produce platelets. And in essential thrombocythemia with calreticulin mutation, this is what the bone marrow looks like, packed full of these megakaryocytes making too many platelets. And this is what the therapeutic antibody does. It completely clears them away. It's incredibly exciting, I think. And they showed that it had this effect on the disease-causing stem cells, also very exciting. So, you know, that's where I wanted to end, really. This is a super exciting new treatment and an amazing opportunity to specifically target mutant calreticulin blood cancer cells. And that is coming to phase 1 clinical trials, so first in human trials this year. And we are fighting very hard to try and make sure that it comes to the UK as well as America. So there we are. Thank you very much for listening. Here's some nice pictures of Oxford, and we've got a nice amount of time to take some questions at the end.
Thank you, Adam. It really does seem as if you're on the cusp of some great changes in how treatment will be, in how the 3 different conditions will be treated, and really excited to hear about the antibody therapy. I've got a couple of questions from a naive point of view. There is a themed question. There are a couple of themed questions coming around trials. One of those I wanted to ask was the JAK2 trials in ET and in MF.
Yeah.
That you mentioned that they're in trial and they're in the pipeline for trials. How close are those trials likely to be available to patients in the UK? Well,
on a trial basis, those trials are actually open.
Oh, right.
They're open in the UK. So there are different sources of information for which trials are open in the UK. And, you know, what tends to happen, like it or not, but this is the reality, is that clinical trials for myeloproliferative neoplasms, because it's a rare disease, they only tend to open up for recruitment in the centers that have got a particular specialist interest in MPN. And so that means you get these little hubs of centers that are running these trials. Oxford is one, Guy's Hospital in London is another. And, you know, there are various different trial centers, usually in the big teaching hospitals, kind of dotted across the UK. When we run a bigger trial, like the MAGIC study, that was actually open in 30 different centers across the UK. So it was open in many, many centers. But often these trials with these new therapies, particularly when they're run by a drug company, they only open up in 4 or 5 centers across the UK. But those studies, the phase 3 studies, are open, or some of them are at least, and recruiting, and you know, they're going to report some of their findings hopefully towards the end of the year.
So leads to answer the question in chat, really. And
yeah, exactly.
If somebody's wanting to get involved and find a trial, what's the best route for them?
Well, speak to your local haematologist and ask whether they know about any clinical trials. And sometimes if you're in a region, if you're looked after in a regional hospital, that may require them to get in touch with their kind of tertiary referral teaching hospital center. If you're not having any luck, there are some kind of web portals that can be used that have information about trials. For example, the MPN Voice website is a useful source of information for clinical trials. And, you know, if you just Google my name, you'll find my email address, and you can drop me an email if you're stuck, and tell me a little bit about where you're based, and I can put you in touch with a haematologist locally.
That's
but the first port of call is with your local haematologist.
Excellent, and thanks for putting yourself out there as well. I can add to that as well that at Leukemia Care we have a patient advocacy officer. Our advocacy officer is there for you to contact, and the advocacy team can also help you with connecting and talking to the different healthcare professionals and what's available to see if they can connect you. I'm going to ask you a naive question, because I'm not from this, you know, I'm a cancer patient myself, and a chronic cancer patient. So I understand some of the differences between treatments of fixed duration and driving treatments into a deep remission and the molecular response that you're talking about. And there's this new treatment, this targeted antibody therapy, which looks as if that could be a massive game changer, being cancer specific.
Yeah.
So there are two questions that are going to come out of this. But in achieving the deep remissions and the molecular response, are these all continuous therapies, or are you looking to be able to approach an era where these are treatments of a fixed duration? And is the new treatment, the targeted antibody therapy, is that a continuous therapy, or is that a treatment of fixed duration?
Yeah, I think we're still in a phase in myeloproliferative neoplasms where we're looking at continuous treatments. Okay. Now, you know, obviously the ultimate goal here is to have a treatment where you give a fixed treatment duration, eradicate all of the blood cancer cells, and then the treatment can stop. That is definitely the ultimate goal, but we're definitely not there yet. So at the moment, I think all of these trials would typically continue to administer the therapeutic agent whilst a patient is responding to it.
And would that include the latest development with the targeted antibody therapy? As that's got a purpose of eradication.
That's primarily preclinical at the moment. So that's moving to phase 1 trials at the moment. But I doubt they would, at least initially, put in a discontinuation kind of module into the trial. I mean, if you look at chronic myeloid leukemia, another form of leukemia where there was a hugely effective on-target treatment available for patients, it actually took quite a while before we started running studies where we tested how safe it was to actually stop the treatment. So, you know, we've got to take things carefully, of course, and one step at a time. The risk of discontinuing the treatment is always, of course, the disease might stop growing back, and then if it does grow back, maybe next time round it won't respond so well. So my sense is always to be cautious, continue the treatment if patients are getting on with it and they're responding well. And, you know, in due course, we can, you know, if we're being really successful and really inducing these molecular responses, then the next question is, I agree, how safe is it to stop the treatment?
Okay, so that leads me on to my next question. Please, people in the audience, if you've got any questions, because obviously you're living with the condition, and obviously some of the news that you've heard, you might have not fully taken on board, or want to ask Adam questions, please stick a couple in chat. It leads me on to the next question. So what this all means to patients is potentially treatments achieving a deeper remission and a deeper response, complete response, and molecular responses, which mean living with fewer side effects and being side effect free from a disease point of view. What about the treatments? Because you mentioned earlier, and again, this is me being naive with ruxolitinib, potentials for shingles reactivations.
Yeah.
So you didn't mention how that's managed, and I'm just wondering, are there any, with these new treatments, what are the side effect profiles like of these new treatments? Yeah. Because living on a continuous therapy, a little issue over a long period of time isn't a little issue. So I'm just wondering if you've got anything to say.
It's super important. These are the decisions where you have to make to, as doctors together with the patient and their family, all the time. This is how medicine works. You have to balance the potential benefits of a treatment versus the potential risks.
Absolutely.
And there are no treatments that are completely side effect free. And that applies to all of these treatments coming through. They've all got potential side effects that have to be, closely monitored for. So, as the agents are developed in clinical trials, we obviously learn what the important side effects are and what we've got to watch out for. And when these agents are introduced into clinical practice, we have strategies to try and mitigate those side effects. So, for example, ruxolitinib and the risk of shingles is a good example that's something we've learned about that drug as we've got more experience with it. And, now in patients who have a history of previous shingles, I would routinely put them on a treatment to help prevent a further reactivation of shingles.
So they'd have acyclovir as a prophylaxis that they would potentially...
Acyclovir as a prophylaxis, and there are now vaccines that are becoming available. The important thing about the vaccines is it's important that it is not a live vaccine. There is a live form of shingles vaccine, but there's also a inactivated form that is available, and that's the one that patients who've got a blood cancer should have.
Well, it's all extremely exciting from somebody outside of living with the condition. You did ask, you just raised an interesting question in my mind there, where some of these treatments are myelosuppressive, and you mentioned vaccinations. Are there specific vaccination requirements for people on treatments so that are they as effective if
that's a very good question.
Myelosuppressive treatment?
Yeah, that's a very good question. And we don't recommend in patients who are on continuous treatment that you stop the treatment to receive vaccinations. That's the first point to make. Secondly, probably there is some detrimental effect in terms of the immune response to vaccination if you are on a strongly immune suppressive treatment. But nevertheless, having a vaccination is still much, much better than not having it. And it varies a lot from treatment to treatment. So with ruxolitinib, there is definitely a negative impact on the immune responses that people achieve to vaccination. This has been particularly particularly studied in the context of COVID vaccinations.
Have you got your eyes on chat? We have a couple of questions that have come in, one relating to shingles prevention.
So, yes, I can see a question from Lisa Southall. What are your views on anagrelide and anemia? I have terrible anemia. I now take acyclovir as shingles preventative. I think that was all spelled perfectly, actually, Lisa. So what's my view on anagrelide and anemia? Anagrelide is a treatment, just for everyone's information, that is used typically in patients with essential thrombocythemia, so high platelets, and it is a very effective treatment at reducing the platelet count. However, there is a potential side effect of anagrelide that we know about, that it can increase the risk of development of some bone marrow fibrosis and scarring, and that can also be associated with the development of anemia. So my personal approach, I can't really specifically comment on individual cases in the context of this type of meeting, but my personal approach broadly to the use of anagrelide is to make sure that we carefully monitor what's happening in the bone marrow. So before starting a negride, I like patients to have a bone marrow. Isn't a nice test, I know that, but nevertheless, it's really important to see, is there any bone marrow fibrosis at the beginning before you stop the negride treatment? And then I would recommend every few years, probably every 3 years or so, a repeat bone marrow just to check for early signs of bone marrow scarring, and particularly if there's evidence of development of anemia on the treatment, that might be something to think about. And the other question from Audrey Greenfield was about a negride. So hopefully I covered that in the answer. A negride is a very good treatment for patients with essential thrombocythemia. It is very effective at controlling the platelet count. And in some patients, it is a sensible choice of treatment, and I do use it. However, again, going back to clinical trials and how important they are, there was a very big clinical trial that was run in the UK over many, many years. It was running, I think, when I started off in hematology. That is now quite a long time ago. And what that trial asked is, in patients with essential thrombocythemia, which is the best treatment, hydroxycarbamide or a negride, and patients were randomized to one treatment or the other. And what that trial essentially showed is both treatments are very good at controlling the platelet count, but hydroxycarbamide is somewhat better than a negride at preventing some of the disease complications, such as arterial events such as stroke and heart attack. And a negride was in that trial associated with a slightly increased risk of the development of myelofibrosis. So my personal approach to the use of a negride is, it's not the first line treatment that I would go to. I use it as a second or third line treatment in patients that have not responded to the standard treatments.
Thank you for being really thorough with your answers. And I don't think we've got many more, and I'm noticing we're getting short for time. I guess probably another naive question, from a patient's perspective, people hearing about molecular response and how that's, a measured treatment approach, and wanting to achieve a complete response in terms of an event-free survival. If people are on current treatments not achieving that and want to achieve that, how many of these trials, how able are people able to move to new treatments, and what would be those, what would be the mediator, the mechanism, or the signal to do that? And how many trials will enable that?
Yeah, I think it's a really good question. I will get in trouble if,
I have to ask.
If all the patients on the call go to their hematologist and say, I want to achieve a molecular remission because that's what Dr. Mead said we should be doing. I don't think it's ready for that kind of prime time in terms of how we manage patients. I think, and what I mean by that is, if you're on a treatment for a myeloproliferative neoplasm and you're responding well in terms of good control of the blood counts, symptoms and spleen and so forth, but you haven't achieved a molecular response, I think at the moment we're not quite there yet to say, oh, you need to switch the treatment because you haven't achieved a molecular response, because everything else, control of blood counts and everything else, is good. So it's not quite there yet. It may be something that comes over the coming years, but for the moment, that's not what I would do in my practice. It is something that we measure, and it's a good thing if it happens. But if a molecular response is not achieved, it is not a reason for kind of panic station. That's the wrong word. It's not a reason for, switching treatment, because there are always risks associated with switching treatment. Whenever you make a decision in medicine, as I said before, there are risks associated, whenever you introduce a new treatment. So that would be my answer to that. It's looking very interesting. I think the molecular responses are important, and it's definitely a good thing to measure, and definitely a good thing if someone achieves that. But not achieving a molecular response is not in and of itself a reason to switch treatment. It's got to be taken in the mix of all the other important parameters that we measure.
That's great. I think we've got time to take that last question that's just come in with regards to the CALR trials. I don't know if you can answer that.
So yes, it's a question from John Kearney. My pronunciation of names is terrible, by the way, so sorry if I get these wrong. Hi, in relation to the calreticulin clinical trials, how long do these trials last? When could we expect any recommendations to roll out such a treatment? It's a very good question and a super important one, and we do like to try and move things through as fast as possible. So, but there has to be a careful process, of course. So the process is, first of all, phase 1 trial, where it's really about establishing a dose level that and safety of a particular approach. That would take 6 months to a year to complete that typically. I think it could go quite quickly for this trial because there's so much excitement about it. And once that is done, then typically things move forward to a phase 2 trial where a larger cohort of patients are given the drug at the established dose level to establish the safety and side effects in a larger group of patients, but also to start getting a sense of how effective it is. And if that also looks promising at that stage, and that takes another year, then it's a phase 3 study, which is like those randomized studies that I mentioned, where you compare the treatment against the best available treatment that's out there. That would tend to take a year and a half or so. And once that's done, then the company has to present all their data to the regulatory bodies in the US, the FDA, and in Europe, EMEA, and now in the UK, we've got our own bodies, of course, MHRA, and that process takes about a year. So you're looking, unfortunately, and this is best case scenario, everything going really smoothly, about a 4 to 5 year time span from first patient receiving the drug through to it potentially becoming available as a therapy. And, if I reflected on that process, would it be possible to make that process quicker? To a certain extent, possibly. There is a huge burden of administration that comes with these trials, but at the same time, the trials are so important to ensure patient safety, and that always comes first. What I would say is my own practice is, I run a lot of trials. I've got about 30 trials open in Oxford at the moment. My personal motivation for doing that is that it speeds up the access that patients get to these new medicines and by making the trials available, rather than having to wait until, they've gone through all these steps and then gone through the, cost effectiveness, National Institute of Clinical Effectiveness review and so forth. And we're often the last in the UK to get these drugs approved for routine use in the NHS. So that's why I do trials, to get access to these drugs more quickly. The final comment, I know I am waffling on a bit, but the final comment is what companies often do when they've got a very interesting and exciting new treatment is whilst they're waiting for those important regulatory approvals, they make drugs available on what's the old-fashioned term is a compassionate use program. Now it's called a named patient program. So the JAK2 inhibitor treatment I mentioned, momelotinib, that is now owned by GSK, GlaxoSmithKline, and they have got a compassionate use program available in the UK now for patients with myelofibrosis. And so the drug company provides that free of charge to patients. And so that's a route to get some of these drugs a little bit earlier. And we do a lot of that in Oxford.
That's great. I think we've just about run out of time, and we'll draw a line under things there. Really exciting listening to this. From a patient's point of view, it sounds as if these developments are coming to a head, and the trials are going to start to becoming more available, which will be a route to access in the interim until things go through the regulatory process, and maybe if there's early access schemes with pharma through pharma before routine commissioning. So an exciting time as a patient looking forward to living with a chronic condition it's amazing how time does pass. And what sounds like it's a long way away soon arrives at your doorstep. So it's really uplifting to hear, and I've learned an enormous amount. I've got a few slides to share just as we part, and then we can part for the weekend. If I can just find my slides, and as usual, I have a problem unable to find my slides. Bear with me a second, please. Sorry about this. It's a Friday, fingers sticking. Okay, there we are. Thanks for everyone for your patience. I'm having a cursor problem today. There we go. I'll just click my way through those. So those who are familiar with Leukaemia Care will already know of many of our services, but we do provide quite a wide range of information sources. So webinars, we've got a continuous stream of webinars. So keep your eyes on the website, keep your eyes on information. Register with Leukaemia Care, and you will receive, the regular magazine and newsletters that will keep you up to date. We also offer a podcast service for information. We're very active on social media, but if you haven't visited the website, take a bit of time and have a good look through the website to see what you can find. Booklets are are available. We've got a wide range of books, ETPV and myelofibrosis treatments and management. So you can visit those and access that through the website. Support services, the helpline is always there for you. And if you're not somebody that likes to chat on the phone, then there is also a WhatsApp route to the helpline. Virtual support groups I'll come on to in a second. We have a national network of support groups, if you're not already aware of them. Some of these disease-specific, local or national. Welfare advice is really important at this time. Everybody's struggling a little bit in the financial climate, and there are grants and ways to actually help, as well as the advice. So our welfare officer is always there for you for a call. Advocacy support I mentioned earlier, if you're needing help trying to look for clinical trials or, to understand some of your own situation and communicating with your doctors, please do contact us and speak to our advocacy officer. Sophie's a wonderful person. She's got lived experience herself, conditions, and she's there for you. Online forums, we're very active on Facebook and other areas, and I know many of you will also be using external MPN groups. Always a good idea to mix and match. The buddy scheme I'll talk about in a minute. That's a great way to meet with other people if you don't get the opportunity, those that actually understand exactly what the situation is. And we do offer a counselling fund. Living with continuous therapies, living with these cancers, this can be a challenge, and the emotional and psychological impacts, there is a route to help you with that. We do offer a 6-session counselling fund to help you actually gain counselling if you'd like to get counselling. And I can attest myself, there have been times I've taken counselling going through treatment, and I stole the virtues and benefits of that. Booklets I've talked about. What seems to have happened, I think I've gone up rather than down. Here's an idea of some of the national meetings. Wednesday, 6 p.m., 10th of May, there's the PV national meeting. I'm sorry about using the acronyms, because I would certainly struggle with all of the new treatment names that you've just gone through today, Adam. And again, 24th of May, there's the ET national meeting. And I think there's a myelofibrosis meeting that's probably later on in the year. But if you look on the website, you'll get an idea of exactly what's there, and there might be something that fits you. Buddy scheme I've mentioned. We've got fully trained buddies, and we've got a really extensive buddy network. And if we haven't got a buddy for you at the time that fits, we'll go out and look for that person for you and bring them up to speed to work with you. So please get in touch if you want to have somebody that you can chat to on a regular basis. That can help support you. Please do. Advocacy case where I've spoken to you about a welfare officer. Slides here just for the review. I've also mentioned about the counselling fund. Here's our number to be able to contact us for anything. Just leaving that on the screen for you and for the recording whilst I find my cursor. So I'll stop the share here, and I'll use this as an opportunity just to say, Adam, thank you. You are wonderful as ever. I don't know how. I mean, it's so complex. I thought, I mean, I'm a different type of patient in the chronic lymphocytic leukemia space, and I thought there was a lot going on in our space, but it's certainly not as diverse as in yours. But thank you so much for the talk, and I hope everybody in the audience has found this of great use. Thank you for joining us on a Friday. It's now the end of Friday. It's the start of the weekend. So have a great weekend, and hopefully this rain will stop. So,
have a nice weekend, everyone.
Thank you very much. Bye-bye, everybody.
Bye now.